Welcome to Botany. Today we are dealing with the topic ABC model of flower development. This model explains how meristem developed into floral meristem then finally into flower and why it has been given this name. This ABC are class of homeotic genes which interact together in the development of flower. And we know the homeotic genes are genes that governs the development of anatomical structures of plant. And also remember, in the case of animals, it governs the body plan and structures like we have Hox genes in our body. Now getting back to the meristematic tissue, we know initially it is the undifferentiated cell called meristematic cell. Then after the differentiation, it differentiates into three types of meristem. 1 apical meristem, 2 lateral meristem and 3 intercalary meristem. Furthermore, this apical meristem differentiates into SAM, shoot apical meristem and RAM, root apical meristem. It's from SAM which give rise to floral meristem, and we see here it is the ABC class of homeotic gene which kicks and in turn shoot apical meristem into floral meristem. Now let's try to understand the flower development through diagram. In the structure of flower, you can see the flower has got four important structures. Outer wall is the sepal or calyx, then it is followed by the petal or corolla, that is the colored part of the flower. Then we see stamen or androecium, the male part of the flower, and the last innermost part of the flower, carpel or gynoecium, that is the female part of the flower. We designate this part as 1, 2, 3, 4 from outside to inside as in whorls. First is the sepal and the innermost part is the fourth whorl, that is the carpel. For understanding the floral diagram easily, we can represent the floral whorls in concentric rings. The outermost ring designated by the first whorl sepal and followed by the second, third and fourth whorls. So this illustrates how flower organs are developed in whorls. Now we will try to explain the homeotic genes for the flower development. We know there are three classes of genes that aid in the development of flower. A class genes, B class genes and C class genes. The A class genes comprises of two genes AP1 and AP2, that is Apetella 1 and Apetella 2. Then B class gene comprises of two genes AP3 and PI, that is Apetella 3 and Pistillata. And lastly, the C class genes has got only one gene by the name of AG or agamus. And it's by these five genes AP1, AP2, AP3, PI and AG genes that interact together and transform the shoot apical meristem into floral meristem. Class A genes, that is AP1 and AP2, promote the development of sepals. Also, Class B genes, that is AP3 and PI, promote the development of petals and stamens. And finally, Class C genes or agamus gene promote the development of stamens and carpels. In order to understand the gene contribution for the floral structures, we will use the concentric floral diagram. In that diagram, we see class A gene contribute for the development of sepal, and then both A and B interact and contribute the development of petal. Then the third structure, stamen, is promoted under the expression of B class and C class genes. And finally, the carpel is promoted by the expression of class C genes, that is agamus. Then, let's make generalized gene interaction diagram for the floral structures. First, we have class A genes. Its expression develops the sepals. Secondly, we have class B genes. As we know, B interact with A also and promote petal development. Then we have class C genes which interact with B also and develop the stamens. And finally, C class genes individually promote the carpel development. So, this is the generalized gene interaction for Arabidopsis thaliana. From this diagram, we can see A and C are mutually antagonistic to each other, and also B genes do not express individually. That is, B is expressed either when combined with A or C. A and C antagonistic means if A is mutated, C is expressed, and if C is mutated, A is expressed. Now let us discuss about Mattebox genes. The Mattebox is a conserved sequence motif. The genes which contain this motif are called as Mattebox gene family. The floral homeotic Mattebox genes such as agamus and deficient participate in the determination of floral organ identity according to the ABC model of flower. What is the function of Mattebox genes? In plants, Mattebox genes are involved in controlling all major aspects of flower development, which includes male and female gametophyte development, embryo and seed development, root, flower, and fruit development. Now let's get into the mutation part. In order to understand the mutation phenotype of flowers, we will mutate the A, B, and C genes and see the phenotype obtained. First, if A is mutated, that time B genes will be as such. Also, there is no change in the C gene expression, but the A gene is not getting expressed here. So, the C gene will be expressed since both are mutually antagonistic. So, in place of A genes, we have C genes. Now, in the development of floral structures, we see the C will develop the carpel at the first whorl. The second whorl should have B A plus B, but the mutation of A causes it to become B plus C, which develop into stamen. Then the third whorl will be B plus C, the stamen, and the fourth whorl is C, that is the carpel. So, after mutation, sepal and petals are replaced by the carpel and stamen. Furthermore, we can look into the C mutation. Since C is mutated, A is expressed in place of C. Here the A will be giving us sepal and A plus B gives petals, B plus A gives petals and A gives sepals. Here the third and fourth whorls are replaced. In B gene mutation, the generalized gene interaction diagram will be without B genes, since B genes are not antagonistic to any genes. So here in this case only A and C will be expressed. Firstly, A will develop sepal. Secondly, since B is mutated, only A is expressed, which will develop second whorl into sepal. Similarly, third and fourth whorl will be carpels. In this type, petals and stamen are replaced by sepals and carpels. And if all the 5 genes are mutated, then pseudo-flower development will occur. That is, a green leaf-like structure is formed. So, these are all possible phenotypes when A, B and C genes are mutated. This is all about ABC model of flower development. I hope you like this video. If you like it, give me a thumbs up and make sure to subscribe this channel. Thank you.